project led by the Prince William Sound Science Center. Our partners include the Alaska Sea Grant Office, the Prince William Sound Audubon Society, who hosts one lecture a month, and also the United States Forest Service. So our lecturer next week is going to be Hannah Johnson from the Alaska Department of Fish and Game. She's going to be talking about the subsistence surveys that were done here last summer, which is part of an ongoing project to monitor subsistence use in Exxon Valdez oil spill affected areas. So that's going to be there. She's going to be talking about the data that they've collected and kind of hoping that we can give her an idea of whether it looks right. So I'm hoping lots of people will show up for that. And this week we have Russ Hopcroft from Fairbanks, and he's going to be talking about the research, Gulf Watch research along the Seward Line. Welcome, Russ.
Okay, so feel free to interrupt me at any point if there's something I'm not making clear. I have no place else to be tonight, but I've targeted this for about an hour. So, but you know, if it takes longer, that's not an issue. So I'm the front person for work that actually represents the efforts of many people. And in addition to the many people, this project has been running for 18 years, and I have a lot of different people to thank for supporting it. Primarily, this started out as a National Science Foundation, NOAA collaboration, and it turned into one that was heavily supported for about 5 or 6 years by NPRB and North Pacific Research Board. And now NPRB, along with the Alaska Ocean Observing System and the Exxon Valdez Oil Spill Trustee Council through the Gulf Watch program, are the major contributors to this program. So, starting at the basics, what we're talking about when we want to understand the oceans is that transfer of energy that comes in from the sun into the oceans, and that energy is what is taken up by the algae that live in the ocean. And those algae can then turn around and it can fall to the seafloor and support animals that live there, or it can be transferred into the small organisms, the drifters called zooplankton, that live in the water column. And from there it gets up to things that we see more readily, such as forage fish species, and they in turn get eaten by bigger fish that we would actually want to use commercially, or eaten by seabirds or marine mammals. And of course they get eaten by people as well. So plankton are essentially the link between the sun's energy and all those organisms that we normally think associated with the sea. And in particular, were there no plankton, there would be no fish to harvest in most areas of the ocean. So we have this pyramid of energy that goes from the smallest things, and many of them, and becomes increasingly concentrated as we move through the food web. This is sometimes called the food pyramid. And at the top we put charismatic things like marine mammals, or perhaps in Alaska we should be putting the salmon up there on top. But what accounts for the changes in these populations over time, we believe, is largely climate. What a year is doing different from a year before or after. So that climate variability is transferred through changes in the ocean's physics and the ocean's chemistry, and they, from the bottom, push at this food pyramid and determine how it's going to be different from year to year. Oh, wrong way. So if we really want to understand what's going on in the oceans, it's not just enough to study water temperature and to study the top. We need to study all the components of this pyramid to try and figure out the way things are working their way through the system. Because we can't just connect climate variability to the top predators, to things like the fish or the commercial harvest, if we don't know what's going on in between. So as an ultimate example of why we care, one of the big changes that's happened in climate in the North Pacific is this change in atmospheric conditions, a condition called the Pacific Decadal Oscillation. Went through this big change that changed the overall structure of the ecosystem in the mid-1970s. And it changed fisheries in Alaska, in some areas, being dominated by shrimp, which primarily are feeding off energy that's fallen out of the water to the seafloor. And so the food's all on the bottom of the ocean, to one that is based more on production that's captured inside the water column, up high, and transfers through the plankton and through small fish into the top predators. And this is the kind of change some of these areas underwent associated with this change in the climate. So it's not just an academic exercise, it's an exercise that has commercial impact and required retooling of an awful lot of the fisheries in Alaska to change what was being harvested. So to make sure we're all on the same page, because I can't help myself in what I call some things, I want to just remind you the terms you're going to see repeatedly. We're going to talk about phytoplankton, which is the algae, the small plants that live suspended in the water. Phyto means plant, plankton means drifters. They are actually not eaten first by something we can see, but something we can't see, which are small, single-celled predators that are also eating the single-celled plants, and then both of these compartments are eaten by the multicellular plankton, which ranges in size from something of a grain of sand all the way up to something like a big jellyfish that's tentatively as big as I am in terms of what weight. So the dominant zooplankton in the oceans are actually types of crustaceans related to shrimps. The thing on the top is called a copepod. It's the most abundant life form in the ocean. The one that you're probably more familiar with is krill or euphausids. They're the things that are more like an inch or two in size that we know we'll find in fish stomachs. They look like a shrimp. But there's a variety of other diversity in the plankton. These are both herbivores as well. This is a pelagic snail or a pteropod and a sea tadpole. And these are both grazers that are quite common in the Gulf of Alaska. And then we shift into the things that are more predatory. We all know that jellyfish eat things. They eat other plankton generally and small fish. They range from being just a few millimeters in size to several meters. And there are other predators that we don't know much about in the ocean, such as this arrowworm, which has got these big hooks on the end of its head. It's a darter. It grabs prey and then it continues to push it down its throat. And these actually go from about an inch up to about 6 inches in size in some areas of the ocean. So the area that we're going to be focusing on tonight is the North Pacific, in particular the Gulf of Alaska. The Gulf of Alaska is separated by the North Pacific Current into a gyre or circulation cell that is kind of north of the Canada-US border. And then that also splits to the south of the warm water current that goes down the lower 48 and feeds into California. So the organisms that live in this upper part, we characterize them as subarctic. They like cold water. And the areas to the south of that generally prefer warmer water. They're temperate species. So up at the top of that, I'm going to be talking primarily about the Seward Line today, but I will, toward the latter quarter of the talk, start showing you some examples of what we're learning about Prince William Sound. So I'm going to make it a little bit more relevant near the end. The Seward Line is this big red line we see right here. And we have been doing since the beginning. We started out sampling this part of Prince William Sound, usually because storms blew us off what we were trying to do, and we had to find some place to hide. And over time we realized there were very few cruises we didn't have to go hide, or we got our work done and had days left over. And then in later part of the study, we've expanded these stations over here, and we now do the western side of Prince William Sound as well. So, right here at this anchor station here at Gap 1, we have about 40 years of data on temperature and salinity and some of the physical processes. Biological data started about 18 years ago, where we started looking at a full ecosystem approach. This started with the National Science Foundation's GLOBEC program in 1987 to 2004. And then I've been begging for money ever since to try and keep the time series going. NPRD, as I said, was a big contributor, and now I have a consortium of 3 primary agencies that keep it running. And in GLOBEC, we used to go out 6 or 7 times a year. I didn't have a life, and now we've reduced it to just twice a year. We have a cruise in early May, and we have a cruise in early to mid-September. And this expansion that I showed you here, all these sites kind of got added in starting in 2011 when EVOS and Gulf Watch came to the table and were kicking in some of the money. So what do we do when we go out? We measure the physics. The physics are measured by electronics sitting here on the bottom of this cage of instrumentation. They measure temperature, salinity, conductivity, oxygen, how much light is transmitted through the water, and a whole bunch of other things at very great accuracy. We have bottles that we close electronically that are sitting on top of that up here. These bottles, we close them at depths we choose to in the water column electronically, and from that water we determine the amount of fertilizer or nutrients in the water. We look at the different forms of carbon to get at the question of ocean acidification, and in some of the studies we've actually measured the concentration of iron, which we now understand in the Gulf is an important micronutrient that sometimes is what's limiting how much this phytoplankton or algae can grow. Oh, wrong button. From the bottles, we also take samples out and measure chlorophyll, which is the pigment that makes plants green. It's a quick and simple way for us to get an estimate of how much of this algae is in the water. And in some cruises, we've actually measured how fast that algae grows. From the bottles, we also measure and count and identify some of the phytoplankton, as well as those very small single-celled zooplankton that are collected. And then we use some very expensive and elaborate butterfly nets, effectively, to pull them through the water. You got a question?
For chlorophyll, could you define how exactly to get chlorophyll? Because isn't chlorophyll attractive to sunlight, or do you get chlorophyll from the actual animals?
Yeah, we take the water and we filter it onto a glass filter, and then we put it into acetone, which is a solvent and dissolves everything. And then we measure the amount of chlorophyll that's in that solvent in a machine that's very, very sensitive for low concentration. Basically, it flashes light at them, and we measure how much light's flashed back at a different color. Okay? Good question. Okay. So for the multicellular zooplankton, because they have very different sizes and very different potential to avoid capture, we have several different butterfly nets that we use to collect them. And to try and make a better connection to higher trophic levels, we actually have people out in the boat who are counting from the flying bridge during the days seabirds and marine mammals. So we can correlate how the plankton is correlated to the things that might be feeding on them. In a perfect world, we'd also be doing fish trawls, but that requires a very different kind of research vessel than what we have access to, and more time that we don't have the funds for right now. Luckily, NOAA does an awful lot of fishery surveys in the area, and we can leverage on that. So let's kind of start at the beginning in terms of the cycles that are driving this. It's a very, very seasonal system, as you're all aware. A lot doesn't happen during the winter. A lot starts to happen in the spring and through the summer. So in the spring, as the light starts to come back, this is what the algae would need to grow. And the problem, though, is that there's lots of light. We know that it's stormy during the winter and the early spring, and that algae is being mixed through the whole water column and actually spends very little time in the layer where it gets enough light to grow well. So one of the ways we stop mixing and trap that algae where it can get a lot of light during the day is we stratify the water column. Stratification means that it has different densities or different buoyancies in the water column, and the algae that's trapped in the more buoyant layer gets trapped in the layer where there's lots of light. And we can make that water more buoyant by warming it up and making it lighter, or by having fresh water come out from things like the glaciers that helps make that water float on top of the other, traps the phytoplankton near the surface. And what we find when we look at it, it's probably not a big surprise for those of you here, Prince William Sound actually starts this process of blooming, of the algae taking off and escaping their predators before the rest of the shelf does. So here we see a picture in early April. We can see this is a satellite picture, and the redder it gets, the higher the concentration of chlorophyll. And we can see that these high concentrations are here in Prince William Sound, and they're in this coastal current that runs along the shelf. The rest of the shelf here is stratifying later. It actually has to wait for the water to warm up enough to create that stratification because it didn't have a lot of fresh water on it. So when we look across all the time that we saw in Golbeck, May is the time, more or less, when the phytoplankton has this big jump and where it increases in its biomass by an order of magnitude, tenfold, and it then feeds this ecosystem for a large part of the year. Everybody just jumps on it and feeds on it as much as they can. And it's not that it disappears in the summer, but it's more tightly controlled by the things eating it during the summer. So, well, there's not a lot of it. Oh. Well, there's not a lot of it in the summer. There's a lot of primary production, a lot of growth going on in the phytoplankton. So, to kind of walk through what's happening in terms of the physics, this is temperature, the colors, and this little strip chart here is showing you the month and the year. And this is showing you what typically happens here in the Gulf of Alaska as we go through the year. The winter starts out, the temperature is the same all the way through, it's mixed by storms. As we go into that late summer, you notice how stratified and warm that layer just on the surface gets to be in the Gulf of Alaska. And this is what's driving the seasonal cycle of the organisms and the production and everything that's happening in the Gulf. At the end of the summer, we start losing heat from the ocean. There's no more heat coming in from the sun. It gets stormy again. Those storms mix everything down into the water column, and the production in the system starts to disappear because the light's going away, and it's no longer trapped where there's lots of light. So now if we look at the biology, and we're going to jump to the zooplankton, this is the spring cycle in the Gulf, and it is dominated. Those two big peaks you see in the spring are by one of these three species of this copepod called Neocalamus that are in this ecosystem. So this is the bread and butter food of what's going on in the Gulf of Alaska, are these big copepods that show up in the spring, and they actually disappear out of the system partway through the summer. I'll come back to that in a second. And that is what we think an awful lot of species are adapted to try and exploit in this system. A lot of higher, bigger organisms. What happens later in the summer is we change over to smaller and faster growing species of copepods. And this is an example of one of them. And while this sounds very trivial, and why am I bothering with this? The reason it matters is because these are now all on the same scale. What we're doing is we're changing from a spring ecosystem that's dominated by this neocalinus and this neocalinus to one that doesn't have these and just has little things like this. So if you're a fish and you want to pick the best time when it's the easiest to go out and feed, you want to try and be out there during the spring and early summer before these guys disappear. And these bubbles you see inside of these are oil. So these things, and this guy is really, really loaded with oil. These things are really, really energy dense. And you don't get the same with the things you find during the summer.
All depends on how big a mouth you got, though.
It all depends on how big a mouth you have. It does. So this is what the life cycle of the neocalinus species looks like. And this species spawns it's actually finished spawning for the year right now. They spawned down here 500 to 1,000 meters deep in the ocean a month or two ago. They're all done, they're spawning right now. The eggs that, okay, they lay their eggs at depth, they never come back to the surface, they never feed as an adult. Their juveniles, their babies, grow and float toward the surface and then sit there waiting for the spring bloom to happen. They have a point they can't go any farther without food. So the problem of a life cycle that's adapted or selected to guess at the best time to be in the surface water is you don't always guess right. If you get there too early, then you may starve to death before the bloom actually starts. And if you get there too late, it's like coming to a party late, you know, all the finger food and everything's gone and you've got the leftovers and you're not going to do as well. So the trick here is for it to hit the optimal window and know that it won't always hit it right every year. On average, it hits it right. Year to year, it doesn't. And then once they finish doing all their growth, they actually go down to the bottom. They store up with all those lipids, they go down and they sleep for the fall and the later half of the summer. And then they wake up and they start producing these eggs again. That's the life cycle of this animal. And how long it takes them to complete their life cycle is actually driven then by how long they're in the right food environment. Yep.
In the other picture, I noticed that one of the zooplankton was carrying eggs. So is that just like the eggs in development, or are they being carried to a specific place?
Yep. There are two types of copepods. There are a number of them that spawn their eggs loose in the water and just leave them behind. They make a lot of them. And there are those where the mother invests more and carries a few large eggs with her. And the thought is that the animals that do that, the chance of the eggs being eaten is much lower. So she's putting more energy into each one of the eggs. And both strategies work. But under the right conditions, one strategy works better than the other. Right? So that's what makes zooplankton interesting, because there's a whole bunch of different ways that different animals do different things, and yet they're all successful under certain situations. And that's why we have all sorts of species of zooplankton instead of just one.
Do these neocalinus die a single year life cycle?
Pardon?
So once they get to the bottom, release their eggs, they die?
Yeah, they're an interesting animal because the female in none of these three species feeds. So all these eggs that it produces here in January and February are dependent on the food, primarily the spring bloom that happened the year before, 6 to 8 months beforehand. So in a sense, what we see here is uncoupled or lagged from what the environment was doing that year. Okay, so the starting point in every piece of science is you got a little bit of information, you come up with a hypothesis, you try and describe how you think the system works. And then you go out and test whether or not you were right. And sometimes we're right, and a lot of the time we're wrong. And then we come up with a new hypothesis. That's the way science works. So when I came to this ecosystem, my hypothesis was that in warm years, the bloom happens earlier because the water column stratifies sooner. And Neocalanus should grow faster because the water's warmer. And I would expect to be in a situation where everything's done sooner. And early blooms usually aren't as big, so we thought that it might not be as good a food climate. And that in years where it's colder, everything's pushed back in time. And that means that you expect to have a longer growing phase and probably even bigger animals at the end of the growth phase than what you had in a warm year. And the reason this matters is because salmon tend to be in an environment that's always cold all year round. It doesn't tend to respond to temperature the way the ocean itself is spawning. And the time that they hit the ocean is actually a lot less variable from year to year. So you end up with a situation there where when it's been a warm year and the Neocalanus have done everything sooner, they're actually going to overlap some of the fish that are coming out to feed on them less well than when it's a cold year. So two predictions here. One is how fast Neocalanus goes through the system, and the second is how well they align with a potential predator and how they might affect the success of that predator. So over the 18 years we've been doing the Seward Line, this is what the time series looks like. We have, in terms of average temperature in the water column, we've got 3 years that are really warm, and we've got a period that was really cold and kind of years that have done different things in between. This is the average age of the Neocalanus that we saw on each of these cruises, where younger stages or younger age is down at the bottom and older age is up at the top. This is the Seward Line going from, oh, I'm real trouble with that button.
It happens to everybody.
I keep forgetting to switch to the laser. And this is the northern end of the Seward Line, this is the southern end of the Seward Line. So there's the stage. So we got it right with the stage being driven by temperature. What we didn't get right was we didn't find that Neocalanus itself, how well the population does, was heavily correlated to temperature. We can see examples here of one of our best years is in one of our warmest years, and one of our best years is also in a cold year, and just about everything in between. So we got half of it right. We got the development part right. We got how well the species does wrong. And before I make the next point, I want to make sure we're all on the wavelength. So this is kind of an aside now, and we're going to come back to where I just left off. I'm going to talk a lot about temperature or whatever anomalies. An anomaly is how different something is from the average or the mean. So this top figure here is the real temperatures along the Seward Line, between 7 and 4 degrees. The bottom one is I've taken the mean and I've subtracted the mean from the observation, so that positive values in this are when the temperature is above the mean, and negative values are when we're below the mean, but the overall picture looks pretty much the same. Okay? So when I talk about anomalies, I don't want you to get confused. I want you to realize positive means there's more than average and negative means less than average. Okay? Now, forget I showed you this because the story works better without this slide. But I wanted to make sure you understood where I'm going from here. So, I just showed you that temperature drives neocalinus development, but it doesn't explain the overall success. But interestingly, what happens when we talk about anomalies now, so this means, when neocalinus abundance is below average, pink salmon survival from the hatcheries tends to be below average. And when neocalinus is above average, salmon survival tends to be above average. It's not a perfect relationship. We don't go to the casino and bet on this one. It's a tendency. And there will always be, as biology, there are always exceptions. In fact, this relationship worked really well until I got into the 2010 stage. So I should have stopped and I should have published it then. But what's happened is actually over the last few years that relationship has broken down a bit. And it's broken down because I'm talking about the neocalinus that are there when the salmon hit the ocean. They're only using neocalinus for the first few months of their life. And then they got another whole year where all sorts of other things can come into effect. As well, salmon have got this really hard to understand odd even cycle in Prince William Sound, where usually there's, is it the even years or the odd years are always good, and then every once in a while it has 2 good years in a row or 2 bad years in a row, and that's making this pattern difficult to line up well. And the hope is that over time we're going to see this come out a little bit more strongly, or at least understand why the years don't fit that don't fit. So this was encouraging. We got the development right, and we showed that how well the neocalinus biomass, how abundant neocalinus is, does affect salmon. And here's another example. I've just built one story. I could also build you a story with the sea butterflies that shows that the sea butterflies in the summer are actually correlated with how well the pink salmon do the following year. So I'm telling you one story, and I could tell you a lot more, but I don't want to be here all night. Yes, sir?
Would that be opposite then? Because pink salmon would be some of the predators, so if predators did less well, would that mean that they did better?
When they first hit the ocean, it's a question of how well do they overlap with the copepods, the big copepods in the spring. As we get a little farther into the summer, those big copepods disappear. And then what we think becomes important to them are these sea butterflies, which are more numerous in the summer, and because the other things have gone away. So they change what they like to eat in the summer. Okay? But these salmon are eating all these things. Okay? So that's kind of the first, there's actually 3 sub-stories in here. So the first story is showing the importance of plankton and showing how that can determine something of commercial value in the Alaskan ecosystem. The second part, which is a question that we always ask in ecosystems, is what does a warming climate mean? What does a warmer Gulf of Alaska mean to the ecosystem that's there? So coming back to this graph, which we've now seen several times, we can see these periods of warm and periods of cold, and we can use these warm periods potentially to tell us what a Gulf of Alaska that's continually warmer would mean in terms of the way the animals and the ecosystem responds. So again, if we look at the GAC1, which is the longest time series here, it's at the northern end of the Seward Line, we can see that over the last 45 years, we've had this change from being predominantly blue, i.e. a negative anomaly, to being actually predominantly red, with, of course, periods that tend to cool off in there again. So So the Gulf has been getting warmer, and the last couple of years have been interesting because we actually had something that got named the blob, where there was a big blob of warm water that developed in the North Pacific. And it started in the fall of 2013, and we could really see it strongly when we went out for our cruise in May of 14, although it's a little bit farther offshore. By the fall of 14, it had intensified and moved inshore. It hung around all winter, so the whole winter from September through to May stayed a couple of degrees above normal, a couple of degrees Celsius, which is double that for Fahrenheit. And it hung around even into the fall, more offshore again, and at the same time we had a condition called El Nino, which is a central equatorial Pacific phenomena that we know has big impact on the whole Pacific ecosystem development. So the press, or at least those who were scientifically literate in the press, started arguing about this fight that was going on between the blob and El Nino, and who was going to win and who was going to be more influential in this system. So I've shown you that the blob was influential, and this is the index we use that tracks El Ninos, and these are the 3 biggest El Ninos that we have on record. We're on like number 3 of the ones that have been observed since they started calculating this index 65 years ago. Yep.
Kind of off topic, but who drew that?
Who drew that? I would guess it's this fellow. Babinski? I don't know what newspaper this came out of. I had someone send this to me because it was cute.
So they obviously were aware of the blob and the names that you gave them.
At the public level, it was kind of described as a fight between which of the two was going to be more important. So, and I could have brought in all sorts of stuff about the blob, which was an old Steve McQueen's first big movie, which some of the people in the room will know who Steve McQueen is. So,
Or saw the movie. Or saw the movie.
yeah, or saw the movie. Yeah. I don't think I was. I'm not quite old enough to have seen it at the drive-ins. So, which none of you younger ones know what drive-in is either. So, anyway, so what did this mean in terms of real stuff in the Gulf of Alaska? Here is what the fall of 13 looked like before we saw, along the Seward Line, before we saw the impact inshore of the blob. And we can see this temperature, kind of normals around 10, 11, maybe up into 12, little bits of it. Here's the fall of 14. We're up at 13, 14 degrees, 2 degrees warmer in the surface water. And that persisted in the fall of 15, and in fact in the fall of 15, it, if anything, was even deeper in terms of its impact. And with this data in mind, actually before we had the blob hit so heavily, what we see along the Seward Line is over the 44 years we had data, we can see that there's been, yep.
What is the scale of the digicores? Is that depth?
Yes, that is, pressure and depth are the same in these units.
So how many feet roughly?
It's decibels of decibars of pressure.
Meters.
So a decibar, of pressure is the same as 1 meter. Sorry. I keep forgetting, depending on who prepares these figures, it helps me prepare these figures. The physical oceanographers like to work in pressure because it's a real thing you can measure, and they have to calculate from the pressure. They make a few assumptions and then get depth in meters. So they're smarter than me, so they like to work with pressure, and I like to work with depth. So, anyway, what we've got now on the Seward Line and the GAC-1 stations, we can show, in fact, that what we're seeing here is part of a long-term trend. That the Gulf of Alaska temperatures have been increasing, salinity has been decreasing at the surface, which means there's more fresh water, more warm water on the surface, and that salinities in the deep water are actually increasing. And we think this is going to start to play out in the way the ecosystem works and the way that the bloom and timing happens in the spring.
So how about the deep water? Is that staying fairly constant, or is that warming up too?
Royer showed that there's an increase, Tom Royer, a retired professor, showed there's an increase in the deep water temperature as well. And that's a global phenomenon. So that deep water temperature has been increasing globally just at a slower rate. And now we actually, with the Argo strictures out, we are able to get a much better handle on how that deep water's responding. So I showed you some averages of the surface. Here's what it now means in terms of anomalies. So we can see this really strong, this whole shelf in the fall of, in May of 15 is hot by a couple of degrees, and particularly the fall before in 14 and persisting through 15. And we're really curious to know whether this thing has fallen apart over the course of this winter, or whether we're going to see it again when I go out in literally a month from right now. I'll be at sea looking at what this year's doing. Yeah.
Is the salinity depend on temperature by any chance? Because I had known about this, but for the end of May, the temperature was very high and the salinity was very high. So they seem to have extremely...
There is a relationship between temperature and salinity, but they can do different things. So if you take the same piece of water and you cool it down, it becomes saltier. And if you warm it up, it becomes fresher. But the kind of changes I'm talking about are driven by adding fresh water in. So you can add fresh water in and make it fresher, not just by temperature. Is that clear? Okay. So as a biologist, well, it's interesting to see what the temperature is doing because we expect it to have some kind of a biological effect. This is the biological effect. So this is the average abundance of 3 copepods that don't really belong in the Gulf of Alaska. These belong in the California current system, the south here. So in, I never think I'm dyslexic until I do something like this. So we actually saw for the first time, we saw these warm water species actually show up, or near the first time, in the spring. Because it was so warm over the winter of 14-15, some of them were actually able to hang out and not be killed off by the winter in the Gulf of Alaska. What we're used to seeing is we're used to seeing them show up in the summer, and in fact some of the highest concentrations of these species have been in these last 2 years where we've had this really, really warm fall. Now, before you get all alarmed, this is a signal that we can detect, but this is not a wholesale change in the community. These are only a few percent, I think a maximum of 5 to 10% in this species here in the whole plankton community. So we didn't replace the whole community, but we had them added to the community that was there. But what we don't know is if we continue to warm up the Gulf of Alaska a couple more degrees, or we keep it warm for a whole bunch of years in a row, we don't know to what extent these southern species will actually become more and more important. And we also don't know what the species that normally live in the Gulf of Alaska, what their kind of upper limit is before they tend to suffer from this warming. So these are the things that we're kind of looking for in the future. So part 3, now, if there's any questions on part 1 or 2, I gotta jump in. Part 3 now is to try and relate what we're learning on the Seward Line to closer to home for you, which is inside of Prince William Sound. So the Seward Line sits over here, relatively speaking, and this is Prince William Sound, and you can see the coastline outline. And the point I want to make here is Prince William Sound is really, really deep. Before I came up here, somebody said you've got a fjord that's deeper than the shelf. I'd say, oh no, you're pulling my leg. This end of Prince William Sound, many of you probably know, gets 800 plus meters, so 1,600 fathoms deep. This whole shelf is the result of glacial sculpturing from the last and previous ice ages. And you can see all along this coast these recessional moraines deep here in the ocean, a couple of 100 meters deep, from where the glacier pushed out through Resurrection Bay here. And there's the glacial moraine. So this whole shelf is really, really complex. And Prince William Sound is this deep probably because there's a fault there and because the glaciers just gouged that out completely as they were being pushed through these narrow passages in the Sound. So the interesting thing then is you've got really deep water trapped here that we don't have a connection to the deep water that's out off the shelf. You've also got an ecosystem that is fed by an awful lot of really, really cold and fresh glacial water that is different from the habitat out on the shelf. The shelf's got a lot of small fjords, but they don't have any comparison to the size and influence of potentially keeping a distinct habitat together inside of Prince William Sound. So I just wanted to make that point before I go farther. In terms of the physics then, what this sets up is a situation where here's the temperature along the Seward Line from last fall. And I want you to realize that, well, it's got a lot of warm water on the top, just like Prince William Sound does over here. So this is the Sound, this is the Seward Line. There's a lot of cold water at depth out on the Seward Line year-round. This is the water that never gets warmed up by the sun. That really cold water is absent from the deep water in Prince William Sound. Literally from about 200 meters down, the temperature doesn't change in the deep passages in Prince William Sound, which I find is really strange. The second thing that doesn't happen is on the Seward Line, as we get into the far offshore oceanic water, salinity continues to increase the deeper and deeper we go. That doesn't happen in Prince William Sound. It caps out here. You can see this 33. This line we see here is now way down here in Prince William Sound. So Prince William Sound is also a little fresher on average at depth than what the outer shelf is. And the last thing that's unique about it is the North Pacific is well known for having what's called an oxygen minimum zone. By the time you get down to about 500 meters off the shelf from the Gulf of Alaska, there's almost no oxygen left in the water. And organisms like us and fish need oxygen to survive. That oxygen's gone because the North Pacific is so productive that stuff falls down and rots. Bacteria eat the oxygen up. You get to a point where all the oxygen's been used up. Prince William Sound doesn't have that, which means that there's a lot of fish that could live at depth in Prince William Sound that couldn't live at those depths out off the shelf. Yeah.
White boxes.
Well, that's no data. So that means that I think I forgot to add when I created that. I didn't have bathymetry, bottom depths, to put into the Sound. So there should be a gray area there that shows where there's a shoal. And this is actually starting here at Montague Strait and running up around the top of Night Island and Naked Island and coming back down here to Hinchinbrook Entrance. So this is a big loop through the Sound. But to me, the interesting thing is most of the Sound actually looks pretty much the same through the whole domain.
But say what you just said again about the fish and the oxygen, that in the Sound then, that they can survive better, or just that they're going to stay there?
Well, it means that oxygen won't be determining the potential depth of fishes, of deep water fishes in the Sound the way it could be offshore.
Oh, okay.
So I guess one way you could look at it is that in theory, a halibut could probably go down to the bottom of Prince William Sound and not suffocate. I mean, that'd be a way to look at it. Maybe that's why some of the areas around here are really, do well in bottom fish. Whereas if you fish the same depth offshore, you might find that there aren't fish there because there's no oxygen for them to live in. To me, it's just an interesting phenomenon because it will determine the distribution of a lot of zooplankton and probably fish. Okay, so what I want to do now is ask the question of how does the zooplankton differ between the Seward Line, where we have all this work, and Prince William Sound. And to do that, I'm going to need to use some sophisticated math and techniques. Now, I'm not a great mathematician, so I'm using software packages that know how to do this math. I'm not doing it myself. But what I want to do is I want to determine how similar one jar of plankton is to another jar. And then once I figure out how similar all these jars are, I want to look for the pattern in how these jars, how their similarity compares to their position in time or their position geographically. Does that make sense? Okay, so the method I'm going to use actually accounts for the absolute abundance of the different plankton species, and it accounts for their composition. And most of you know how to think in like 2 dimensions, so if I write on a piece of paper, that's 2 dimensions. If I look at something, that's got shape to it, it's 3 dimensions. This analysis creates as many dimensions as there are species. So this analysis, if I have 60 species, is working in 60-dimensional space. Ooh, it's kind of like a blob all over again. And then what we try to do once we figured out where they are in this multi-dimensional space, we try and figure out how close each of these pairs of samples are. And then we start taking the ones that are closest together and then grouping them with the next closest and the next closest. And we literally build a tree that tells us about how similar they are. We can show that as what looks like a tree with branches, or we can show that by taking all these dimensions and squishing them down to 2 or 3, and then plotting them spatially. So here's the one approach where I've created this tree, and what it says is that these samples, when these joined, these samples were 90% the same, effectively, and when they joined up here, they were 78% the same. And what you can see is there's some major groupings in these samples. This is all the data from May in the Seward Line and Prince William Sound from 1998 to 2012. And when I try and figure out where these different groups are, I end up with this cluster over here that's really distinct from all the rest is Prince William Sound. So this is showing us that Prince William Sound is a slightly different zooplankton community from the rest of the survey area. The second cluster is actually the entrances. So this is deep Prince William Sound, and those are the entrances of Prince William Sound, where this Alaska coastal current kind of peeks in through the mouth of Hinchinbrook Canyon and flows along the back of Montague and then out through Hinchinbrook, or out through Montague, and the very coastal stations along the Seward Line. And then the rest of this is the farther offshore water along the Seward Line. So this analysis is showing us that there's structure and that there are differences between Prince William Sound and the Seward Line proper.
Oh, where was the other shoe? You left that group off to the right, just hanging.
Oh, what I've learned, having done this analysis a lot, there are always going to be some samples that just don't behave themselves. Right? These are ones where somebody spilt half the jar and didn't want to write it down because they didn't want to be held responsible for it, or something really weird was going on with the sample in terms of the way it was preserved, or that just an eddy or a jet or something had brought a different kind of community into that sample. And I've learned to, rather than get concerned about this, because we would always go back and check the samples, and the data, it wouldn't be that we recorded it wrong. So I think it's biology. Biology has chaos thrown in on it, just to keep me employed. And you had a question?
It was just about it was about the soil.
About the extra, yeah. So these are the misbehaves. I call them the black sheep. Okay? And every time I've ever done this, it's very rare to not end up with a couple of black sheep. But they're always, I do in fact go back and check them, because every once in a while I find out that somebody wrote something down wrong. That this was, because we take only a piece of the sample, sometimes they said, the records say we took a quarter, or we really kept like 100 and 1/64th of it, and that can be enough to make them fall in or out of the clusters. And sometimes, so different things can happen in a record keeping. So it's a cross-check. When it happens, we check it. If it's real, and we can't mark it. So this is the other analysis now, where I take all those dimensions and I just squash them down into 2 dimensions. I can show you this in 3 dimensions, but until I rotate it in real time, you can't see the 3rd dimension. So, and I haven't figured out a way to animate it yet. So the point here is similar in that here's Prince William Sound, all these yellow samples here. And these are some of the ones that didn't fit from the last diagram that are just out to lunch. So Prince William Sound actually is, it should be, if I showed you this and I showed the 3rd dimension, Prince William Sound should be going like this, not lying flat compared to the rest of it. It should be going in a 3rd dimension. It's that different. And then the innermost stations along the Seward Line are these green stations, GAC 1 through GAC 4. The most offshore are the dark blue, and then we got this kind of transition zone in here that sometimes behaves like it's really offshore water, and sometimes it behaves like it's coastal water. Yeah.
How are some of the Prince William Sounds all the way down there on the other side?
These are the ones that were on the last figure. So I think those are, in fact they are, see these stations here, see those little yellow dots there? It says these are the black sheep
Yeah,
in this diagram,
got it.
and they are the black sheep here too. And I could go back and see what color the other black sheep were. What I do now when I do this kind of analysis, when they're black sheep on one, I mark them as black sheep. I mark them as black dots now when I get to this level. So, several pieces of information are starting to say that Prince William Sound behaves just a little bit different. And as a cartoon, one of the ways we're starting to think about it, this is courtesy of Ken Coyle, is that we've got these species that live in that layer that warms up on the surface. And that layer that warms up on the surface, the wind pushes it around a lot. It moves inshore, offshore. And in fact, we find the species that live in it tend to be the same across the whole domain. We've got another group of species that really are more oceanic species that live in deep water. And they tend to be in the outer end of the line. And in fact, they don't tend to get transported across the shelf. They don't end up in Prince William Sound. Then we've got some other species, midwater species, some of the migrators, that seem to be carried on the bottom water that comes in across the shelf and over the ledges of Prince William Sound and get carried in. And in fact, we find that some of these species actually accumulate in really, really high concentrations inside of Prince William Sound in the deep channels. In fact, they're more abundant in the deep water in Prince William Sound than what they would be in the offshore water along the Seward Line. So Prince William Sound actually ends up being a bit of a concentrating mechanism. And I think the fact that there's lots of oxygen and some different properties in the bottom of Prince William Sound actually makes that a good environment for them.
Is that throughout the sound, or let's say right Montague Strait, the entrance there, Ingeborg entrance?
It tends to be in the deeper water. I'd have to go through and dissect the data a little bit more to see if it's specific to a region.
Mm-hmm.
But it always amazed me when we do the collections in Prince William Sound. A lot of these things that are deep water species actually migrate up and down at night. And we have far, far more of them inside of Prince William Sound than we see out on the shelf ever. And it may be because that water mixes better in Prince William Sound. The channels are really well mixed by tidal activity. or maybe because the habitat is more consistent through the water column. I said we don't get that stratification, that layering of the different types of water in the sound that we get offshore. So to kind of summarize some of the points that were there, I thought it was easier to just summarize than it was to show you a whole bunch of data pictures. So these surface dwelling species that are really, really open ocean, we don't get them inside of Prince William Sound. What we do is we get actually a concentration of species that are really coastal in character. They tend to get concentrated in the surface water of Prince William Sound. As I said, these deep water species can be extremely concentrated in the sound compared to what we see them on the Seward Line. And I think the concentration of some of these species is one of the things that lends to the higher production of thumb stings in Prince William Sound than the offshore waters. There are certainly some species of fish and other animals that do really well in the sound, and animals that come into the sound to feed during the winter. The other neat thing we're starting to realize about Prince William Sound is what we call glacial relics. So if we go back to the last ice age, Prince William Sound was part of the Arctic, as was the whole North Pacific. Covered completely the land with glaciers, and you know it was like the shelf in the Chukchi Sea or the Beaufort Sea or off of Greenland. As the glaciers receded, some species got left behind in habitats that didn't warm up, like Prince William Sound, the deep, cold water glacial fed fjord. So we find species in there that we don't find until we get into the northern Bering Sea, or until we get into the Arctic proper. In fact, there's a species of copepod that we see here called Matridia opotensis, which is named for the Sea of Okhotsk, which is that big sea behind Kamchatka in Russia, which also gets ice covered in the winter. Yep.
How do you distinguish these anomalies from the black sheep?
From the black sheep? The black sheep we only see a few times, and we can't figure out why. The other ones are things that we see again and again and again. So it's a pattern that repeats, and we can find a good explanation for it, or a good story to go with. Maybe it's not really the truth. Okay? And things that don't make any sense at all regardless of how hard we try. Okay? In fact, I had a student who's starting to do genetics now on some of the copepods, and she found one of the copepods there that we normally call an Arctic copepod living inside of Prince William Sound. We don't find it on the shelf. And there's some other examples. This fellow here, this Cyphalocaris challengeri, like we get just full of, the samples are full of them, like hundreds or of them sometimes come up in one net inside of Prince William Sound. We never see them off the shelf. This is normally a very deep water species, and maybe that's why it shows up. We don't get it on the shelf, and we don't see it offshore. It's too southern a habitat for it. And one of the things that Rob Campbell's getting a little bit better handle on than I am is that some of these small warm water species that I say belong in the California current system actually seem to be finding a way to make it through the winter inside the surface waters in Prince William Sound. And I think that's going to be something that Rob and I are going to be scratching our heads over for the next couple of years as we try and figure out, and they seem to be becoming more abundant. And if that's because the water in Prince William Sound doesn't cool off as much as what it does out on the shelf, or that there's some areas that that they like fresher water like they get inside of Prince William Sound, or the food just stays better all winter round in Prince William Sound, we don't know. But this could be becoming another one of these examples of something that becomes more and more common in Prince William Sound. And then from there they wash out onto the shelf. So, there is a fourth piece. How are we doing on time?
It's 4:10, so I think we're good. We got lots of time.
I can show the fourth piece, or I cannot show the fourth piece.
Show it. But before you show it, can I ask a question?
Sure.
About the fall bloom?
Yeah.
You haven't mentioned that at all. is that, I know sometimes in the sound you have a fall bloom, is that, do you get it out there?
We do see often a fall bloom out in the Seward Line. It's even shorter lived often than what the spring bloom is. So when blowback was existing, we sampled in August and October. And the bloom happened somewhere in between there. We never saw it. Now the Seward Line cruises in the fall. This was a logistics thing that happened from 2005 on. We've been in mid-September. So now we often see that fall bloom because we're sampling at the time where it may or may not happen. In that turnover time when storm mixing starts to mix some nutrients up from depth and there's still enough light left for the phytoplankton to do something with it.
So it takes some storms to do it?
Yep. Yep. You've got to get nutrients back into the water to fuel it. There has to be enough light left to take advantage of it.
And then what's the carryover effect? Is there a big impact the next year if you have a fall bloom?
Well, it doesn't matter for neocalamus because neocalamus is already down hibernating for the winter. It matters to things like pseudocalanus maybe and some of the other species where it's one last good kick of being able to feed well for a few weeks to put on some extra fat to take them through the winter. So it may in fact help make them more successful.
But you can't make a correlation, let's say, to fish production or anything from it?
Well, because it's a little bit shorter now. It's about 6 or 7 years shorter a time series if we want to look at the timing issue. The physics we can compensate for. We can kind of project the physics into that midpoint. The interesting thing about the Gulf is, well, I can show you all these year-to-year differences in the spring. By summer, they're wiped out. Summer just looks like chaos in terms of it's always warm by then years are just not as different. So we're still wrestling with that part of the story. Okay. So the last piece is I wanted to show you just the direction that we've been heading in my lab for the last little while. Ultimately, what we can learn is to some degree limited by how fast we can keep up with the samples that we collect and get the information out of them we want. Physics is easy. You walk off the boat with a disk and you've got your data. You have to QC it. It takes a little bit of time, but you really pretty much know what you have with the physics. The biology, we walk off with boxes and boxes of jars that have animals in. Somebody in the lab is going to have to go through and split and count, and we're not going to see all the data for 6 months, 6 to 12 months. So is there a way to speed that up? So what there's been a lot of advances made in computer image acquisition and computer identification systems, the ability for computers to recognize people's faces, and is an example of this kind of technology. And so we asked the question, can a computer actually identify zooplankton so my technicians can retire? And is letting the computer do it, in fact, cheaper than having people do it? Can we do more samples? Can we do them faster, better, stronger? So here's a scan of a zooplankton sample. And now we see that contrast I talked about earlier, where we've got lots of little copepods like pseudocalanus and big copepods like neocalamus. This is the kind of spectrum I'm talking about for sizes. So the approach basically, and this sample would never work because too many particles are touching here. The computer can't figure out, when things start touching, the computer can't figure out how to identify them. So what we do is we take a sample that's more dilute, we make sure we've got nothing touching. This is basically, it's a scanner. It's like a photocopier underneath water. Goes through, and then the computer goes through, and it extracts these different images like that, and then the computer tries to put a name on each of those images. So the computer takes those and it throws them into the folder that it predicts is the best match based on what we've taught it. And to get to the summary of what did we learn, it holds great promise, which means it doesn't work completely, but it shows signs of working. So the computer can actually separate them really, really crudely. It's a great first sort. It's a rough sort. And it has trouble with all sorts of images that my technicians can look at that same scanned image and they can tell you exactly what it is. So what we've learned, what grade are you in? Are you the 8th grader on here? 6, okay, I was close. So what we've learned is that we can take, the computer really operates as somebody with very little training. It gets it kind of half right, but it doesn't get it good enough for us to do a lot with it. But if we take somebody then and take what the computer's done and we go in and we say, well, that's a mistake, I'll put it in the right place, and that's a mistake, I'll put it in the right place, we can actually very quickly get to the point where we've got usable data. And it does in fact end up being faster than having somebody look at it under a microscope. And the cost saving here is we can bring in a summer student who can do the splitting and the scanning, and then I can save the hard stuff for somebody who's paid better and has a lot more experience and actually separate them into a lot more species than what the computer could ever hope to do. So a computer plus brains, and this is actually showing great promise right now. And there's the hope that the software will actually get better at doing it. So I wanted to show you that because from that kind of analysis we can create maps of the different species, and we can create them over broader domains with many more samples than what I could afford to pay people to do under the microscope. So this is an example of one of the copepods and cruises we ran in the 3 seasons in 2011 and the 3 cruises we ran in 13. And we can see in both years the species starts out at least being offshore, primarily the shelf edge. And as the year goes on, we tend to find them more onto the shelf. And they appear to have all sorts of complications about where they are that we're still trying to understand. More importantly, with this combination of brains and computer, we're able to then turn around with that same kind of analysis I showed you for the Seward Line to look at the difference between Prince William Sound and the Seward Line. And I can now cluster these and color code the clusters, and we can see that in the spring of both years, we can see there's an offshore group of samples, and there's a habitat with a lot of complexity in it on the shelf here, and a very narrow shelf community here. We saw the same in 2011 and 13, shelf and nearshore communities. And these communities, the colors are all the same between all 6 graphs. The spring is very different from the rest of the year. The rest of the year, we tend to see again an offshore community with a little narrow shelf in southeast, a broader shelf in the western Gulf. And the falls look fairly different in terms of the communities between the 2 years. So we're starting to use the computer to allow us to do more stuff and look for bigger scale pattern in the samples. And this is leading now to some plans of trying to take what we've learned along the Seward Line and what NOAA does in their fisheries assessments along the shelf, including the work I started to show you there, and putting it together. NOAA is starting to do a lot more work down in southeast Alaska. They've had a long time series running since I think '89 or so, Kodiak through the Aleutians. And the hope is that we're now in a point where we've got a lot of data spanning a long time, and we can start putting it together and looking for Gulf-wide patterns in terms of the plankton and the ecosystem. There is a website. It's not always up to date. It is more up to date than this figure suggests. I do have, in fact, a summary of last year on that. So if you want to learn a little bit more about what we're doing, it's there. But as I said, I don't always do a good job of being able to post it quickly or every year. So that was everything I had.
Thanks.
to talk about.
Are there any equivalent time series like the Seward Line in Alaska?
The Seward Line is the oldest and most detailed. There are 2 other time series. I mean, there's a time series that's not quite, well, the Prince William Sound work, some of the physics goes back, 20 plus years. And Rob's harvested all the physical oceanographic data that's there. The biological data, there was a C program, and then there was a kind of a pause, and then Rob took over. Rob's been doing primarily the bays, and we've been doing primarily the passages for the last couple of years, but he started up in about 2000-ish. I think he's been doing the bays.
2007. Well, he started here 2007.
Yeah, so that was going, there's a one by Icy Cape by the entrance of, is it Jeff? Or, yeah, that Noah used to run, but they had a very small boat, and usually they get weathered out, wouldn't get the offshore, so it was all inland passages. And there's a continuous plankton recorder time series that started about the same time as the Seward Line, about 2000-ish, that also has a record for just the surface layer, but they have a good coverage for most of those years. So there's several of them, and I interact with most of those groups, and they're potentially part of the synthesis we're starting to talk about as well.
Hey Russ, so February water temperatures in Cordova, the average February was actually warmer than last year, and that was true from Cordova to Kodiak. It was actually quite a bit warmer in February. If you look at water temperatures, they bottomed out in early December, January, and actually started climbing, in January.
Yeah, and the
So
whole
what are you
Bering Sea-
thinking we're gonna find out there when you go out?
My prediction is we're going to have a lousy year for neocalins. I did some extra work. I wanted to bring up sleeping neocalins last September. We went out to the end of the Seward Line. We ran the nets. It should have been full of neocalins, and all we got was dead animals or no animals. And I was really annoyed because we spent hours going through these samples. We were throwing them out as we went through them because there wasn't anything there. And then, like, later that day, I said, oh, I should have kept them because now I have a story about them having done poorly, but I have no data about them having done poorly. So what we learned was we should have made two collections, one to put away right away before they started to go downhill from being kept too long, and then the others for our experiments. So we actually took the samples in Prince William Sound last fall to show what was there, all the way down to 600 meters, as well as the ones for our experiments. And we actually used to sample down to 600 meters from 2001 to 2010. We used to do that at the end of the Seward Line and in Prince William Sound. But as we scaled up all the other work, it became hard to fit those collections in, and nobody was actively working with them. So we stopped doing them, and now we're starting to do them again. So hopefully that'll provide us with some insights. But my fear is that neocalins had a lousy second half of the year, that their summer through fall, and they didn't get enough energy put away, and the water was too warm. When they went down to depth, they just didn't have enough lipid to take them through.
Great, well, thank you so much for coming.
Great.